The main polymer technologies focus on how to actually make polymers. It's that simple and that complicated. Two sciences can help: chemistry and biology. The chemical approach to making polymers is to find a way to repeat the same reaction one step at a time, changing monomers as needed. It starts by covalently attaching the first monomer to a solid support. The monomer is protected and nothing can bind to it. After a wash, a chemical is added to remove the protection, and a second protected monomer is added. A second deprotection step follows, a third protected monomer is added, and so on. The last step consists in removing the polymer and making it available to the world. This Lego building approach is called solid-state synthesis. It is a great technique because there are only few ingredients, purification is easy, and chemical synthesis is fast. However, errors can happen, and there is no proofreading of what is being built. When the polymers are really large and their sequence is complex, only living cells can produce them. The process is called fermentation if microbes are used, or cell culture if cells from multicellular organisms are used. The main disadvantage here is the complexity of the ingredients and the high number of biochemical reactions taking place inside cells. This can make purification difficult and the overall process quite slow. However, biological polymer formation is precise and self-correcting, and this is a truly amazing feature. A final fundamental difference between chemical and biological synthesis is the ignorance or respect of chirality. Chirality refers to the capacity of some molecules to exist in two mirror forms that cannot be superposed. Chirality exists in human hands, but also in amino acids, and only L amino acids are used by cells. It also exists in sugars, and only D sugars are used by cells. Well, chemical synthesis ignores chirality, but biological synthesis discriminates between L and D forms. Of course, chemical and biological production processes are often complementary. Unfortunately, chemists and biologists have little opportunity to explore possible synergies while being trained. One of the important tasks of modern biotechnology is to ensure that chemists and biologists work together.